Now? Yeah. Cool. So yeah, today we are going to talk a little bit about the complex microbiome, basically the bacterial communities that live in different environments, from like free living environments like the ocean or the soil to the ones that are associated with multicellular organisms. And the reason why, looking at or understanding better the organization of these communities is that bacterial communities in free living environments, like for example the ocean, they are responsible for like a large fraction of the biogeochemical processes that happen on Earth, including like carbon cycle and things that are relevant for like warming, O2 production, and carbon sequestration, and all these sort of things. So it's very like a very relevant topic. And the other thing is that the ones that live in association with multicellular organisms, they provide these organisms with, like basically we wouldn't be possible without bacterial communities. Until recently, the people used to think that for each human cell in our body, we had 10 bacterial cells. That estimate recently changed a few years back when some people redid the calculations, and apparently it's like 1.3 to 1. But still, like if you think about it, like more than half of your cells are bacterial cells. And this happens across many different taxonomic groups of the tree of life, and basically the emergence of metazoan organisms are due to these symbiotic relationships. We are going to see some examples of how communities like this are organized, with a particular example of marine sponges, because in sponges they are known to provide like fundamental functions for maintaining health and survival of the individuals, as well as some examples of the human microbiome as well. And the whole point is to try to see how we can use the tools that we have been looking at during this week, but this time applied to much more complex communities than the ones that we have studied so far, right? So basically, the approach here would be to try to see how we can bring community ecology to better understand microbial communities. So basically, the way in which people have started to address these questions is by looking at these microbial communities as they have always looked at other ecological communities. And for example, they look at things like this. So this is an ordination plot of different samples from marine microbial communities across all of the oceans of the world. And this is the kind of things that you use in I don't know if you have already heard this during this series of like different topics you have been exploring, but basically what happens in this plot is that you run any kind of ordination analysis, like for example PCA, and then that classifies the different communities according to the composition of their species. And the ones that are closer to each other are more similar in terms of their composition. And for example, in this picture, we can see that some of the same kind of colors sort of group together in some place. And that means that they are more similar in those particular oceans than in other oceans in the planet. Then the other thing that people normally do when they have this community ecology data is to look at relationships with environmental variables. So in here we have an example of how one of the dimension of this ordination plot of the PC relates to temperature. So it turns out that temperature seems to be like a strong driver of the assembly of these communities. And then here we have another example of how they do this for multiple other environmental variables. So each line going from taxonomic composition or gene composition, going from there to each of the environmental variables here are the kind of relationships between them. And this is the kind of things that, you know, community ecologists look for when they are trying to understand better the composition of their communities and what might be the drivers behind them. So basically, you know, microbial ecology has sort of adopted this way of thinking, and now we can look at these communities also in this way. This might be useful if, you know, if you guys are analyzing any kind of microbial data, and there is a lot of this going around. But in particular, we are going to focus on a few of the metrics that we know so far. So we already know stuff like alpha, beta, and gamma diversity, so I'm not going to go over that again. But another metric that we commonly use, especially to create plots like the one above for, you know, trying to understand how separate, how distant different communities are, is what it's called, in this case, the Bray-Curtis dissimilarity. But in general, any sort of dissimilarity measure. So you try to, starting from the relative abundances or the composition of each of your communities, you try to compare them by calculating some sort of distance, right? Like, for example, you could calculate even Euclidean distances. But the ones that we use more commonly in ecology, and especially in microbiome studies, is the Bray-Curtis distance. And the Bray-Curtis distance is basically 2 times Cij, sorry, 1 minus 2 times Cij, in which Cij is the sum of the abundances of the smaller species, of the smallest abundance of the species that are common across samples, and divided by the total sum of all of the species in the community, all the counts of individuals of species in the community. And that, like, applying this to a pair of communities will give you like a distance, how different these two communities are. And then you can use this information to you know, quantify these kind of differences in community composition. So the distance between points in here is proportional to some sort of metric like that, right? So this is a way of quantifying difference in diversity. And so before we have been talking about the different kinds of diversity that you can quantify, right? Do you remember which one was related to the differences between local communities? Yes, beta diversity. So using that formula, then we can create plots like this. So analyze this kind of information. So in this particular example, this is an analysis of the organization or the structure of microbial communities in the human microbiome. In here, we have again the same sort of ordination plot in which you can see how different colors represent samples coming from different parts of the body, and they sort of aggregate in different places. So here you have samples coming from the oral cavity, then here from the gastrointestinal tract, and so on. And on this first top plot, we are looking at alpha diversity. We already looked at, yep.
Could you please just remind what is alpha, beta, and diversity?
Sorry, I sort of assumed that you guys were listening to my other lectures. No, I'm joking. Alpha is the local, so if you imagine that you had some sort of like big regional pool, and then you have like local communities, in which you have your species here localized in different patches. And so alpha diversity would be the diversity at the local scale, so in your local sample. Gamma diversity would be the total sort of assemble of all the species that you find in everywhere. And beta diversity is like how much biodiversity you are gaining from one place. So how different, what is the turnover of species between local places? And the Bray-Curtis is a way of calculating that. So in this particular case, for example, you know, local alpha diversity here, which is the first plot, would be the diversity at each of these little dots here. That's why these are boxes. So this is the entire universe of your sort of species composition that you have across all of your samples. So this would be like, you know, gamma diversity here. And then each of these is one of the other measures. So alpha diversity across, for example, the different parts of the body. So you can see here that some parts of the body are more diverse locally and some others are not very diverse. But interestingly, if you look at the plot in the bottom, which is beta diversity, the differences between local communities within the same sort of region of the body, it's more or less constant, right? So this sort of means that you have less diversity in some parts of the body, like for example, you know, this is the genital area, than in, for example, the skin or gastrointestinal. But the differences in these communities between different individuals are similar across all these different sites. And then, yeah, the other thing that shows here is also gamma diversity, but across the different, what is the subjects that were exposed to, oh, sorry, I forgot. Let me check. Okay, so yeah, it's the diversity of the samples from within the same individual versus individuals versus the difference between different individuals. Cool. So using this, yeah, as I said, using these kind of things, you can start like you know, understanding better the organization. And if you want to go, but that, I mean, what we have seen so far only tells you like how different the communities are, but it doesn't tell you anything about why are they different, like what makes them different, right? So if you want to look at these things, one thing you can do is to look at the differences in the taxonomic compositions of the different, in this case, host species. So when looking at the bacterial communities within the specific species of multicellular organisms, you can try to analyze which, in this case, bacterial phyla on the y-axis are more abundant in which ones of the hosts, right? So in this picture here, we have on the x-axis the host species, which are marine sponges, and on the y-axis we have bacterial phyla, and we can see the differences in taxonomic composition across each of the species, right? And this information is useful because there are certain bacterial families or taxonomic groups that are known for specific functions. And you know, by looking at this or analyzing the data in this way, you can sort of understand what hosts are more likely to have specific functions that are fulfilled by these bacterial communities. And well, it also tells you about the heterogeneity, right? So for example, one thing that is kind of clear is that this bacterial family is absent from most of the species, right? So it might be of interest to know, why you never see those bacteria in the microbiome. Cool. So we have more or less seen how traditional community analysis in terms of like alpha, beta, and gamma diversity and composition, which is what we have seen so far. I just wanted to show you that for you to see how you could apply it to like microbiome and microbial communities. But now the interesting part is how we can use more of the network approaches that, again, we have been learning during this week to like build upon those analyses and make make more sort of targeted analysis on different parts of the communities that we cannot see only by looking at traditional community and community metrics, right? And in particular, what people do is to create networks of bacterial occurrences within hosts. So here it's an example of an OTU, or a bacteria sponge network, in which I don't know if you can see this, but there are some nodes that are a little bigger than others. So this, for example, these 3 or 4 here, then there is one here, another here. Can you see the differences in the node? Yeah. So those are the host species, and then the small ones are like bacteria. And in this particular case, the nodes are colored by the module that they belong to after running modularity analysis. So we will look at modularity in these networks. So now we have, it's a little bit different from the networks that we have been looking at so far, because it's not really an ecological interaction network. It's just a presence network, in the sense that it only tells you which bacteria are found within each sponge, right? So what we are going to try to look at here is to try to find communities that are more closely similar across different host species. So bacterial communities that are more similar across host species, and what might be the drivers of those sort of convergence in terms of bacterial communities. So once you have a network like this, you can start calculating network properties, which before we couldn't, right? Because we were looking at it from like a community sort of perspective. So one of the network properties that we have discussed over the last few days is degree. Do you guys remember what is the degree of a node? What is it? Yeah. The number of connections. So basically in here, on the x-axis, I have the number of, or the degree, or the number of hosts in which each bacterial taxa or bacterial species, we call it OTUs for operational taxonomic unit, but I am going to refer to that as species. So how many host species, or equivalently how many links each of these bacteria have in microbial taxa have in the network. And then on the y-axis, we have the fraction of samples from all of the host species in which that host has been found, that microbe has been found, the fraction of the samples within which it was found. Do you understand that? It's a bit complicated, but yeah. So when I have, maybe this will be easier this way. So the way in which you usually get your data for this kind of analysis, including the ones for community ecology type of thing, it's that you have a table in which you have like samples here that might come from different host species. So let's say that these 3 first samples come from species X, then these next 4 samples come from species Y, and then these 3 other samples come from species Z, right? And then in here you have the bacteria, so the OTUs that you found in your, after doing, you know, your sequencing and all the bioinformatics and all that. So with this information, basically you have the, and these are counts, so these are like very big numbers, basically based on the number of reads that you get from the, sorry, maybe I should have explained how you get to this first. Are you guys familiar with how you get microbial data, or no? Okay, well, I'm gonna try. This is gonna be like a very rough summary, so if you are interested in this, you should, you know, like try to read about it. But basically, you take a sample from, I don't know, a sponge or the water, and then you sequence that. And by sequence, I mean that there are techniques for using like different primers and things like that to identify a region in the genome, which usually is the 16S, which is you know, a region that has been, or that is known, that can be used to identify an organism to the species level. And then once you have that sequence, then there is like a very complicated bioinformatics pipeline in which you start removing sequences that you think are not right, and so on. And at some point you need to like put together sequences that are similar to some threshold. And the threshold that people normally use is 97%, although there are some other thresholds that you can choose. This is sort of arbitrary. And then all of the sequences that are similar to that threshold, they get grouped together in what we call operational taxonomic units. That's why it's not like a species per se, it's just like a collection of these sequences that were similar to that to that threshold, basically. And then from each of those, from each of the samples that you sequence, you can actually count, like, you can count how many of those sequences that you classified as each of these OTUs you have in your sample. And this is where, from where all of these numbers come from, right? And then in each of the samples that you have sequenced, you get a big table like this, which looks like basically a community matrix, because you can sort of make the connection between having different communities and the composition of these communities. So this is usually, again, this is like a very rough and quick summary. And if you're interested in how this is done, you should, like read a little bit. There are many like tutorials online and things like that for this. But the important thing for us is that, you know, this is the kind of thing that we have as our sort of empirical data. The raw data, well, not the raw data, but the data after pre-processing a bunch of things looks like this. And going back to this plot, then, what we have in the y-axis is if I know that some OTU, so in this plot, each of these little dots is one row here, one column here. And as you can see, there are so many because, you know, one of the things about this kind of communities is that you have like a sheer amount of diversity. In the first sort of raw data, before I applied some filtering, we had like around 500,000 OTUs. So you need like a very powerful computers also to analyze this kind of data. So yeah, again, in the y-axis here, what we have is we look at where each of the OTUs is located. So for example, let's say that this OTU is found in only the first 3 samples of sponge X. only the first sample in sponge Y and in none of the samples of sponge Z, right? So if there are like 4 samples here, let's say 5 here and 3 here, this, out of all of the hosts in which this OTU has been seen, it was only found in 3, sorry, this is supposed to be a 0, in 3 out of the 9 that it could be in, right? So that is the fraction of samples across which this microbe has been found. And the interest in doing this is that you can tell how persistent, how likely it is that a microbe that has been seen somewhere, it will be found there repeatedly. So the larger the number in here, it means that it is more consistently found across the host to which it has been seen at. And one interesting thing that emerged from this kind of analysis is that for microbes that are very little connected, so they are only found in 1 or a few hosts, they tend to be found in those hosts all the time, right? So they are very, very persistent in those hosts. Then you get like a group of microbes that are found in an intermediate number of hosts. So they are kind of generalists, but they are not really found there constantly. So they are like opportunistic microbes that they are sometimes there, and they can be in many places, but they are not in all these places consistently. And then we again have a bunch of, well, not a bunch, like a minority, but still like a nice set of like very generalized microbes, which are found in many hosts. And these again are found at high fractions in these hosts. So it looks like bacterial communities within marine sponges are organized in a way in which you have like a core set of microbes that are always found in, you know, whether they are specialists or generalists. And then you have a set of microbes that are sort of moving around in different places. And we still don't know why this is. You know, there are like different hypotheses about what like more sort of opportunistic microbes do in the microbiome. But it's, you know, one of the sort of the things that you can learn from this type of analysis. Yeah.
In this analysis, did you observe like an enrichment of the bacterial families, like opportunistic versus very conservative? Because that could be an explanation of why this kind of pattern is seen.
Yeah, that's a very interesting point. I didn't look at that here, but yeah, we definitely have the data to look at that. So, and this data is available and free and public. So if you wanted to look into those things, it's something that can. I didn't do it, but it's yeah, it can be interesting. Actually, you're giving me ideas. Cool.
I have a question about the analysis.
Oh, yeah.
What are the different colors of the dots?
Oh, yeah,
Black
sorry.
and blue?
Yeah, I should have said that. So one of the particularities of this system, of the sponge bacteria system, is that, well, these things have been co-evolving together and co-diversifying for like millions of years, right? Ever since sponge species emerged. And some of the bacteria, the microbes that are found in these hosts, are only found there and nowhere else in the planet. So they are specific, and not only specific to sponges, but many of them are specific to the specific host, right? Or at least to the specific family of hosts, which is another thing that people look at, like the patterns of co-diversification between the families, like phylogenetically speaking. So these colors here, the blue dots are bacteria that are only known to be found in marine sponges, and the black ones are the ones that are also found in like seawater and things like that. So I think that if we go back here, in here, for example, you have these two last columns here are water and sediment. And you see that, for example, this thing is not found in any of this, but it's found in some of the sponges. And conversely, there are some stuff that is relatively abundant in either water or sediment, and it's not really found in sponges. So, yeah, that's the difference between the colors.
Another question.
Oh yeah.
What's the analysis that you got? How did you get the red line? What kind of fitting is that?
Yeah, that's just a lowest fit to the data. It's just to give like a sort of qualitative visual guidance to the pattern, but it's not really it's not a specific function. It's a free parameter kind of fit. Yeah. Cool. So the other thing that we can look at in networks in general, but interestingly applied to networks of host bacteria, is modularity. So modularity is, are you guys familiar with modularity in networks, or more or less? No? Yeah? Okay. So basically modularity analysis, what it does is it tries to identify or partition your graph into subsets of nodes in which the connections between the nodes in each of the compartments, it's denser than the connections to other modules in the network. So if you think, I'm going to reuse this graph for a different thing, but if we think of our network as this whole thing, and then like different connectivity patterns between different nodes in the network. So if we think of this as the modules in the network, and then obviously there are connections between nodes in different modules. Like, different nodes can be connected to others. But basically it's the identification of these sort of subsets of nodes within which the nodes in here are more connected to the nodes within their same group than to any nodes outside of their group, to other nodes in the network. And the way in which we do that is by trying to maximize that function over there, here, M as for modularity. Where this number here is the number of modules, so it's the sum across all of the modules of the links within that module divided by the total links of the entire network, minus the degree of all of the nodes in that module, minus 2 times the number of links across the network squared. And the problem with this is that to be able to calculate that, you already have to have the partition. So you need to know how you assign nodes to different modules, right? So this is like a very hard kind of computational problem, because you need to find the optimal partition in your graph to maximize this quantity. Does anyone here do like graph theory like mathematically, like algorithms for understanding network properties in mathematics? No? Okay. Well, this is kind of one of the traditional kind of classical problems in graph theory, to try to find partitions that maximize this quantity. And there are like many different algorithms around to do this. But one of the ones that is like more efficient are doing this, or, well, it's debatable, but that can do this anyway. It's it's an algorithm called NetCarto, which is based on simulated annealing to make these partitions, to choose these partitions in a way as to maximize this number. I think I put the, I put the reference there in case you are interested in the specificities of the algorithm and if you want to try it yourself. It's also available in R and many different languages, so you can, as a library, so you can just use it like that. One interesting thing about this kind of way of calculating modularity, or well, in reality in any way, but one thing that the people proposing this algorithm came up with as well, is that you can quantify these other two metrics here, which can be used to understand better the position or the role of a node in the network. So the first quantity that we have, which is this little set here, it's called the within module degree. And the second quantity that we have is called the participation coefficient. So basically these things are ways of quantifying how well connected each node is within its module and how well connected it is to modules outside. And then with these two quantities, you can basically draw a plane like this of P versus Z or Z versus P. And by partitioning this space into these 7 different regions, you can come up with like discrete roles that nodes can take in the network. So nodes that have, for example, a very small within module degree and a very small participation coefficient, it means that they probably have one or very few links, and they are only connected to one other node within their module. So they don't have a lot of connections within their module, maybe 1 or 2. And they don't either have like connections to other modules. So they are within their module and they are very loosely connected. And then you progress across this space and you might get to, for example, region 7 or role number 7, in which the participation coefficient is very high and the module within module degree is also very high. So these are nodes that are connected to everything everywhere, right? So they are like super connected nodes. And they are hard to even classify within a module because they could be practically in any module. And the interesting thing about being able to classify nodes in this way is that then you can identify nodes that might perform specific functions within your structure. So like, for example, they can be connectors, even if they are not very, very well connected, they can be connected between modules. And these kind of things have been shown to give you clues about, for example, which part of the network or which nodes are able to connect different parts of the network that might be otherwise isolated, right? And one way of using this information is by looking at, for example, in our sponge data set here, microbiome sponge, we have here, again, the participation coefficient and the within module degree. And then in here, I'm plotting different species of sponges of the host in different types of hosts in different colors. And we can see that the blue colors, it's not important for now what is blue and orange, but what is interesting is that the blue species tend to be like very, very connected to many other species, which means that, and across all of the network, which means that the bacterial communities that live in those hosts are probably shared amongst many other hosts. Whereas the orange type of species, they are very connected within their module, but not very much like to the other part of the network, which means that for these host species, the bacterial communities are very specialized to them, right? And again, this is some information that if you remember the sort of things about alpha, beta diversity, and we cannot know this from that kind of analysis. So this is the key, the sort of fine detail that the network approach can give you on these communities. I mean, just to explain what is the difference between these two colors, because this difference is purely biological. It doesn't have anything to do with the structure of the network. Yet, a pattern in the structure emerges between these two things, right? So, just to give you a little bit of background of marine sponges. Marine sponges, depending on what is the abundance or the density of bacteria, when you look at samples in the microscope, they are usually or can be classified in two types: high microbial abundance and low microbial abundance. And these two things, which well obviously refer to the fact that one have more density than microbes than others, it's also related to different physiological and functional traits, in the sense that high microbial abundance sponges, they tend to pump less water than the low microbial abundance. So they tend to keep their microbes because they like petting them. You know, they like cultivating them. Whereas the low microbial abundance, their strategy is to pump a lot of microbes through their system and grab many opportunistic things. So if you think about these things together, then when we have patterns like this, it's not very surprising, right? Because here you can have microbes that are coming like through the pumping rates of the sponge, and this can be very opportunistic. So you can start creating a picture of like different things from all of these bits of analysis to better understand your microbiome data. So yeah, the other thing that then you can do with, once you have this information about the modularity, is to try to better understand the potential mechanisms behind that. And when we did this, we found that the phylogenetic signal of the sponge species, of the host, it's very related to the modularity of the network. So it looks, and again, this is sort of in line what we were discussing before about the co-diversification of the species, because it means that, for example, all of these orange species, which belong to the same module in the network, they sort of are in the same, not only in the same part of the sort of phylogenetic tree, but they share the same bacterial communities because they are in the same module. So the important thing to remember about things like this is that here we are mixing the structural properties of the network, which were calculated independently without any knowledge of the ecology of the system, and the phylogenetic background of the species, right? So this is, I mean, you couldn't have anticipated this without, that knowledge. So, and I think that that's very cool. So yeah, this is just a way of trying to explore the potential evolutionary mechanisms behind this organization. So the next pattern that people usually quantify, especially exclusively in bipartite networks, is what we call nestedness. Have you guys heard of nestedness? Do you know what it is? No. I think actually somebody asked me that question in the first lecture, and then I said I was going to answer at the end, and I never did. So this is so basically nestedness tries to quantify. If you think you can think of nestedness as a way of knowing, for example, how the subset of different communities, or how different communities relate to each other in terms of their composition, right? And in particular, is a measure for understanding to what extent the composition of a given community is a proper subset of the composition of a community that has more species. So if you have, again, going back to a table like that, if you have your data on community composition and you have here your communities, and here you have your species, then a completely nested matrix or a completely nested communities would be one in which in community 1 we have all of the species. In community 2 we have this. In community 3 we have this. 4, 5. So all of the species in each of the less rich communities are a proper subset of the species in the more more richer communities, let's say. And if you think about this pattern in terms of networks of the kind of things that we were talking about in the first lecture, like plant-animal interactions, if you have your incidence matrix in which here you have the plants, so these are no longer communities but plants, and these are no longer species, but, well, they are species, but they are a specific type of species, the pollinators. And these are the links between them. If you think about this as the diet of pollinators on plants, then nestedness will be quantifying the extent to which the diets of more generalist species, the diets of more specialist species are proper subsets of the diets of more generalist species. Do you understand that? All good? So one interesting property about this, when thinking about networks in particular, about this pattern, is that this pattern has been linked to the stability of ecological of bipartite networks of plant mutualistic interactions. So when communities that show this pattern of nestedness are more likely to be stable than communities in which these connections are like randomly placed. But for now, we are not going to look into these kind of stability things here, but we are going to look at the patterns of this nestedness in microbial communities. So nestedness is calculated using, there are like different metrics to quantify it, but this is an example of one, which is called nestedness based on overlap and decreasing fill. And the decreasing fill part means or relates to how these rows and columns are filled in the matrix. And basically it's a measure of the n paired, which is a number that quantifies the overlap between the diets or the composition of the different communities or the diets of different species in the case of a network, divided by this term here, which is where n and m are the number of rows and columns respectively. So it's like a normalization term based on all the possible diets that a species can have. So using this kind of analysis, we can then look at patterns of nestedness in our case, our microbial data, microbial community. This is data coming from a different system. It's not anymore the host microbe thing. This is actually data from many different systems across the world. And in each of these plots, so here it's all the samples that they had across many different systems and habitats. The first one here is the plant species. No, these are the plant species, sorry. These are the animal species. So these are microbiomes found in animals and plants. And these are two environments, more saline environments and non-saline environments. And here you can see this kind of thing that I was drawing here in the board, right? You see this kind of arch-shaped in which most of the presences are concentrated in one part of the matrix, and the rest is like totally white, which means that in this case, since we have on the x-axis here a specific animal species or plant species or location in the planet, and in the y-axis we have the composition of the community in terms of bacterial taxa, then you can see that communities where all of these bacteria are present contain all of the communities in which the following bacteria, contain all of the bacteria that are present in the community next to it, or nearly all of them. And these guys also did some kind of analysis here in which they tried they sort of simulated the same data but randomizing the presences of the different species across samples, and they quantified the same nestedness knot metric, and you can see that the ones observed in the wild are considerably more nested than the others, which means that, you know, it's probably a pattern that is derived from some biological or ecological mechanism. So what these kind of things tell us then is that we can expect that, you know, if you know the composition of the richest community, then you are probably likely to predict the composition of other communities that might be less species rich. And the other sort of, I mean, I like to think about these things in terms of ecological processes, because I guess I'm an ecologist. So if you think about this, one potential explanation for these kind of things is that these communities seem to be organized in a way that they have like a core microbiome in which those microbes are very generalist and found across many different environments, probably because they are able to do any kind of metabolic functions. And then as you specialize into particular niches or particular environments, then you start gaining microbes that you need to live there, right? So, for example, going back to the ordination plot of the human microbiome, probably the reason why they are so different is because in different parts of your body, you need to perform different functions that for which you need a different set of genes to be able to do them, right? And this is, you know, like, I think at least that the interesting thing about these kind of metrics, that they allow you to start creating this hypothesis of why this is happening, like what might be the mechanisms behind that. And I have always liked the kind of niche theory kind of way of thinking, and I think it's a nice way of thinking about this. So yeah, again, this is just to illustrate that when you are doing this kind of network analysis, it's not just about you know, quantifying the patterns and yeah, we have this. It's also about thinking what are the implications or the potential sort of processes driving it, right? The other thing that we found in I am not showing this here, but the other thing that we found in that host marine sponge paper is that those modules that we found in the network, which again are purely structural and they don't have any ecological information whatsoever, we obtained functional data for the host, for the bacteria that were in each of the hosts. And those modules were significantly different in terms of the functions that the bacteria, that the OTUs provide to the host, meaning that those communities probably were assembled in a way as to provide specific functions that the hosts need, that are different from the functions that other hosts in the network need. And again, this kind of thing, we could only arrive at that by looking at at the network structure of those communities. Cool. Do you have any questions so far? No? All good. So yeah, I guess the last thing I wanted to talk about is, now, again, going back to the general theme of this week, which what I have been trying to do is to show you how you can bring data and modeling together. Because it is important to develop the kind of theoretical models that you guys like, and you have fun drawing equations and all that. That's nice. But then, I think it's very important that we try to bring somehow some empirical data to validate the models. And so now that we have patterns like that, so we now know more or less how the bacterial communities are organized, then I think it is interesting to start developing theory to understand what might be the mechanisms behind that. And this is like a recent sort of, it's a developing field, let's say, because it hasn't been, people hadn't been developed theories about specifically bacterial communities which have their own kind of specific processes and mechanisms. And what is basically happening is that people are adopting the traditional ways of thinking in terms of theoretical frameworks that we have to think about ecological communities. And in particular, the Lotka-Volterra model here. I don't know if it's, maybe this image is a bit small, but you can look at it on your screens. So what people are doing is now using a very simple kind of Lotka-Volterra model, like the one we saw in I think, session 2 or 3. Try to think about the mechanisms that hosts might use to control their microbial communities, and try to incorporate these mechanisms in these models, right? And this is a very interesting way of thinking, because essentially what you are doing is taking this kind of network dynamical approach that has been used for other systems, and try to make it a little bit more specific for the processes that have been observed in host microbiome systems. So what these guys did here in particular, well, this is just an illustration of the whole framework. So here they have like a bunch of bacteria, and they kind of try to predict how they interact with one another. Then they create these networks which are modeled dynamically like this. And then they perform some kind of stability analysis based on the eigenvalues of the Jacobian matrix, that the normal local stability analysis that we use all the time in food webs and ecological networks and all that. So that is not like very super novel. But the novel part is that they wanted to explore how different strategies that the hosts use to manipulate or to structure their microbial communities can have an effect on the stability of this theoretical system. And in particular, the mechanisms that I like the most, because they showed up here as relevant, but they have also been shown in other systems as to structure, as to facilitate or improve stability, is the spatial structuring of the communities. So when you have spatial heterogeneity in the distribution of species, then systems tend to be more stable than in a well-mixed case, because they cannot interact with whatever. They can only interact with the ones that they have nearby, right? And the other sort of host strategy that they tried, one of the others they tried here, was to simulate the targeted sort of the supplementary feeding that some hosts sometimes give to their bacterial communities. And the reason for these two things is that, these kind of systems are known to be very unstable when you incorporate mutualistic interactions because of positive feedback loops and so on. So there is a suspicion that mutualistic interactions are probably not very common in microbial communities, or if they are, that might be modulated by the hosts. And the other thing that has been, and the way in which you can do that is, or incorporate that mechanism into this model, is by decreasing the strength of the mutualistic interactions. And similarly, the way in which they did this, or you can do this in this kind of models, is for the spatial structuring case, is to decrease the competitive interactions and the exploitative interactions. So the like type of predator-prey kind of dynamic, right? So if you modify these terms here, and also this one for competitive interactions, then you can simulate or incorporate these sort of host-driven mechanisms that can make systems more stable. And they did that here. Let me see if we can, oh no. Do you know how to put back the, I don't use Windows, so I don't know. Oh, I closed it. Oh, sorry. Oh, cool. Thank you. Thanks. So yeah, and they did this using not only that model, but they also tried it with an individual-based model to see if those patterns would emerge from local interactions. And here is what happens when you have when you apply spatial structure, but also when you change this PM here, which is the proportion of mutualistic interactions. So it turns out that, in a system like this, mutualistic interactions actually are detrimental for stability. But if you start incorporating spatial structure, that sort of alleviates the problem, right? And similarly, if you have some sort of targeted feeding, which means that you are adding a new term to the Lotka-Volterra equation here, adding a new term for each of the microbial species as a supplementary feeding from the host, you alleviate competition for resources between the microbes in the network, and this is also good for stability. They quantified stability as this local stability thing, and then also as permanence, and also as permanence in the IBM model. So yeah, that's pretty much the way in which you can, if you are interested in modeling, and again, this is kind of a relatively new field, so we are still, I actually have a project going on these things for bacterial communities and how evolution also modulates a little bit the ecological responses. But yeah, this is like a first step towards that, and I thought it might be interesting for you guys to see how you could do this modeling. And at the end of the day, the hope, or not the hope, but the aim of this kind of things is to try to recreate patterns like the ones that we see in these kind of things, right? So. And yeah, I think that's pretty much it. I just, I have a few references here in case you are more interested in other stuff. And yeah, I guess that I finished earlier today. Do you guys have any questions or?
Yeah, I do. Do they suggest any mechanisms by which the host could modulate spatially the communities of bacteria or something like that, or how do the actual mechanism in which the host specially feeds bacteria or something? I just can't connect.
Well, the host just produces metabolites that different bacteria with different kind of sort of metabolic abilities can use, right? So because the bacteria can take resources from the environment. So in the case of sponges, for example, or even the humans, like the bacteria that live in your gastrointestinal tract, for example, they are just exposed to constant fluxes of things passing by, and they use that. And the spatial structuring basically refers to the fact that these bacteria live in the surface of the host, and in different parts the composition of the specific bacteria that are in those parts is different. So bacteria that are here do not like compete or whatever with the ones that are here. Yeah.
How would the host control that? Like which bacteria is going to be here? That's best for me. Which is
it's not so much as control, but just as the body plan of the species. You can imagine that the species sort of developed. For example, in the sponge, the sponges have like specific mini structures, like little cavities, in which the bacteria live. So this could be a consequence of different bacteria colonizing different parts of the body.
Plants deliver some interference RNAs to control the pathogenicity of their holistic bacteria. They have like these kind of channels, and they deliver small RNAs interference to control the invasion, because it has to be slowly. And it's something about related to immune systems in the plants, that it's more like that.
I have two questions. I think the first one is very simple. Do you have a preference for R or Python for working with these networks? And if there are like tools available in both languages?
Yeah, no, not really. I don't. I just used R for this because the projects that I have going on with mostly ecologists, it's easier to communicate in R. But there are libraries available for network analysis in any language. So the iGraph library, the one that I use here, is available in C and I think Python as well. And in Python, there is one called NetworkX, which is also like, it has basically the same kind of functions and like tools to analyze networks. So I don't really have like a preference. In general, I think that Python is way more efficient than R, but I guess it's a matter of taste, yeah.
And the second question is, I like very much the idea of no budget science.
Of what, sir?
No budget science.
Oh, well, I don't know what that is. What is that?
It's like doing science without money.
Without money. Well, how do you pay the bill? How do you pay
I mean, in the sense of using a data set that you can find that was used for something else, and then you apply that like network.
Not to do field work.
Yeah.
Or experiments.
How much this is possible? How common is that?
Well, nowadays there are like databases of everything you can imagine. Like even. I mean, obviously, you usually have to wait for the people who collected the data to publish it. But for example, the data set I was talking about here about the host microbiome is publicly available for the sponge microbiome. And I think that in general, many of the data from the Earth Microbiome Project, EMP, if you search for that online, you can find, I think that in general, most of this data is also available. So yeah, I think it's very easy and feasible to do that nowadays because people they just share the data. Well, a lot of journals also like ask for the data to be shared. So after that, then it's public domain. Yeah.
So like in your works in particular, you generally use like new data, like data that.
No, I have. No, not really. I have always used data that is already available. I still usually, when I develop like a new collaboration, I sort of pair up with the people who collected it, because they usually have like more detailed knowledge about, you know, the ecology, the biology of the system, and they might have also like more specific or more interesting questions about that specific system. But yeah, the data, I guess I had never thought about that, but I guess that, yeah, for most of the things I've done, that I could have done it without, like, just taking data by myself. Yeah. Yeah.
Thank you very much.
Yeah, no worries.
Thinking about having insights about the mechanism behind this relationship, like the microbiome, do we have a better approach using generalized biological Volterra or individual-based models? Are one of them better in giving us these ideas?
Yeah, well, I generally use either one of the other, depending on you know, the question that I am trying to answer. So the times that I have used individual basis, because I am interested in understanding how like individual to individual interactions or behavior affect the system. And also when I am interested in modeling space explicitly at the local scale, not at, like, for example, a meta community or meta population level. So in those instances, I have used individual based models. As I said, when it's relevant, when you think that the behavior of the individuals and the interactions between the individuals cannot be captured by those high-level equations. So in those cases is where otherwise it's probably easier just to go for, you know, like equation-based approaches. Yeah. So for example, for the Tasmanian devil example, that is a nice example, because there are particularities about the way that they interact with each other that would have been harder to capture in equation-based approaches like this. So, yeah. Cool. We still have 14 minutes. You can ask me more questions, even if it's not about this presentation in particular. No? Yeah.
About yesterday's class, I was thinking about, do we have any type of huge initiative to figure out the global ecological network, like just some project like the Human Genome or the Blue Brain project that it's for neuroscience? Do we have this kind of initiative in network ecology?
No, I have not seen something like systematic, like within the same project. What I have seen in those directions is that different sort of big labs, they have collected different networks that they have, you know, that there are out there in the literature, or that they collected themselves, and they put it in like the same kind of sort of platform. So there is this page called the Web of Life, which is like a compendium of different types of ecological networks across different places. There is also the Interactions Web Database, IWDB or something like that. And then there is GLOBI, GLOBI, sorry, which is also like a kind of interaction. That is not like networks. Networks is just like more like two species interaction kind of thing. But you know, you could build networks out of that if you knew like the species that live in your ecosystem. So yeah, I am not aware of any like large-scale project collecting specifically for that project data across the on ecological networks across across the world. But only these sort of initiatives to bring together data from different places. Yeah. I don't know if maybe Pamela, are you aware of? Okay, cool. Okay, cool. Thank you very much for letting me talk to you over these last few days. It was really nice, and I hope to see some of you anytime soon after. Thank you.